Good morning, ladies and gentlemen. I am Belle Shonai, the managing editor of MycoAsia. Today we have Professor Udeni Jayalal, one of our co-founders. He'll be conducting an online training on identification of lichens. And I request our editor-in-chief, Professor Nalin Vijayawardene, to introduce the speaker to the today's audience. Professor Nalin.
Yeah, thank you so much, Dr. Shenoy. It is my privilege and great honor to give a brief introduction to our speaker today, of Professor Jayalal. Actually, I should say that he is one of my teachers when I was doing my bachelor's in Sri Lanka. So, as an introduction, so Professor Jayalal got his Bachelor's of Science degree in botany from one of the prestigious universities in Sri Lanka, University of Peradeniya. Then he started his PhD in Peradeniya, but his most of his lab works has been carried out at National History Museum in London, UK. And then he started his postgraduate from one of the universities in South Korea, Sunshine National University. And he has a forestry master in forestry from Jawaharlal University. So he has different experience, different uh, expertise from different fields actually. So besides, he has published more than 50 research publications related to lichen taxonomy and lichen bio monitoring and lichen secondary metabolites activities. Apart from that, he has been working as a review of many international reputed journals. He obtained several research grants to work on cryptogamic species and flowering plants in Sri Lanka. So you can understand the broad range that he has been working at the moment. He participated as an invited speaker and guest of honor in many national and international symposia. Currently, he is working as a full professor at the Sabaragamu University of Sri Lanka. So, Dr. Professor Jayalal, so it's up to you now. And I should say that he's one of, as I told, he's one of my teachers, and now we are closely working together, and we initiated some works, especially not only mycology, lichenology. We started several works related to mycology. So, Professor Jayalal, it's up to you now. I would like to welcome you here.
Thank you very much, Dr. Shonoi, for inviting to this type of workshop actually regarding the lichen identification as the founder of the MycoAsia journal. And thank you very much, Professor Nalin, for the great introduction about me. So with that, I will start my talk actually. Basically, I'm going to discuss about I mainly focus about the audience that they know little about the lichens. From that point onward, I am going to discuss this talk, and with that, I will move on to this presentation. So, how we can identify the lichens. So, as Professor Nalin mentioned, currently I am working in the Department of Natural Resources, Faculty of Applied Sciences, Sabaragamu University of Sri Lanka. So, with that, now this is the first slide I want to discuss with you. Now, when you look at this slide, many of you may confuse with which part of the lichen in each slide and what are the other organisms you can see. Right now, basically, when you go to the field, you have that knowledge. Without that knowledge, it's a bit difficult to identify organisms even in the natural environment. Now, if I take this first slide, now you can see there are different colors available in the image, even in the other images also. But the most of the parts are in green color. But apart from that, you can see some other grayish or the little bit greenish organisms available in these each and every photographs. Now those are actually the lichens. Now as an example, if you take this one, now you can see this grayish one, this is the lichen, and all the other components are many of the other cryptogamic species like mosses. And when you come to this one, this is, I mean, a bit difficult to identify. Having a bit knowledge, you can identify this somewhat greenish one. I mean, you can see some of the lobes on this picture. So those are the another type of lichen. Actually, this lichen almost, I mean, considering the color, those are almost looks like the other moss species. Now only difference is that the morphology, that means the appearance. You can see this is a kind of thallus-like structure, but others are the mosses you can see in this image. And this is another type of lichen. And this is the one, again, you can see another type of large lichen we call foliose lichen. I'll come to that point later. So this is, you can easily identify even this image, as well as if you go to the field, you can easily distinguish which one is the lichen and which one is the other organism. And even this one also you can see, this either sides you can see lichen, two lichen species, and in the middle part it is a kind of moss species. Now basically how you can identify when you go to the field, one character is that you can use some of the color changes. So that is one basic feature that you can identify. Without having proper knowledge, anybody can distinguish, okay, this is the lichen and this is the moss and so on. And if you have further clarification, or if you need further identify actually is it a lichen or not, then what we can do is we can crush the thallus. Actually we have to go to the laboratory, or using your handless, even in the itself in the field, what you can see, I mean, it's rarely difficult to see whether any chlorophyll or the algal part on the lichen using the your hand lens. But if you go to the laboratory, if you crush or if you cut into your lichen, then if you observe them under the microscope, easily you can identify in the lichen component, it mainly consists the algal part and the fungi partner. So that is the next step that you identify whether it is a lichen or not. So basically you can identify which is lichen, which is not. Then further, if you want to identify which type of lichens that you want to study, then we have to have some thorough knowledge. So here I am going to discuss very basic techniques or the characteristics of lichen where you can use to identify the lichens. Now these are some of the different types of lichen every day you can see everywhere. That means if you are in a city area, most of the time you can't see this type of large lichens. But if you are in a somewhat remote area, you can see this type of very luxurious growth lichen. But apart from that, you can see some of the crustose lichen even if you are in the city, but in the most polluted areas you can't see this type of beauty on the lichen. So I'll come to that point later, because most of the lichens are very sensitive to the environmental pollution, so that accordingly most of the sterile lichens are present in that polluted areas rather than unpolluted areas. And as I mentioned, whenever you go to the forest, or when you go to a nearby, I mean your land or any other place, you can see this type of lichen. Some are present on the tree trunk, some are on the twigs, and also some are present on the leaf surfaces, and also some on the rock surfaces, and even also some of the structures where those man-made structures also you can see lot of lichens on such a surfaces. So as I mentioned earlier, now we know basically lichen is a kind of symbiotic organism where you can see both fungi and algae or cyanobacteria part. Now in this image you can see how they form actually in the nature. Now this green color one is the algal cell where it is wrapped with fungal hyphae. Then you can see a formation of lichen. So I mean, without appropriate lichen and fungi meetings, then there is no any proper formation of lichen. So I will discuss later how they form in a natural environment. So actually recently scientists found that apart from I mean basically algae and fungi, scientists found some other organisms also involved to form the lichen. Now this is one of the recent example where scientists found that apart from this algae and fungi, they found another fungal species, or the yeast species. So once they present only in this lichen, they form this type of lichen. In another study, actually I have not included in this slide, but some scientists found some bacteria and some diatoms also present in the lichen. Actually they help to form the lichen thallus. I mean, somebody will think that other organisms use the lichen as a substrate to survive on the lichen, but actually that is not. Why scientists prove that because of the presence of other than algae and fungi, some other organisms also involved for the formation of lichen. So when come to the classification of lichen, I will first go through this small theory parts. Then after that, I will move on to the other practical components. So the lichen basically composed of algae and fungi. So this classification of the lichen mainly based on the morphology of the fungal symbiont. So that we have to name according to the lichen fungus. Basically we never usually we don't consider about the or the algal or the cyanobacterial component for the classification of the name in the lichen. And this also very important. Basically fungal symbiont I mean never found in free living. However, if you go to the mycobiome part, the other part, that means algae or fungi, they are more present in a free living stage in the environment. But if you go to the fungal partner, it is not like that. And currently there are actually more than 15,000 species of lichenous fungi have been found. And considering all those fungi, many of them are Ascomycetes fungi, and very less amount of fungi found in the Basidiomycetes as well. Now this is one of the example. This what are the Ascomycetes, and they make a kind of fruiting body of the cup-like fruiting body. And even this is the lichen where you can see this brownish fruiting bodies where they produce their sexual spores. And this is one of the example where this Ascomycetes involvement for the formation of lichen. And this is another example where, as I mentioned, the Basidiomycetes also involved for the formation of lichen. Now these are well-known, most famous basidiolichens. You can see how they produce their spores in a it is looks like a mushroom, right? But actually it is a lichen when you identify it further in the laboratory. Now, this is one of the example, and this is one of the another basidio-lichen where they form basidium rather than the ascus. So autotrophic symbiont also very important. Basically, you can identify green algae. The most common green algae is the Trebuchia species, and the other one is the cyanobacteria. Considering the cyanobacteria, the most common autotrophic species is the nostoc species. Even in the autotrophic symbionts, many lichens composed with this type of green algae, and there are, considerably lesser amount of lichen you can see with the cyanobacteria to form the lichen species. Now, as I mentioned earlier, you can see some of the autotrophs in a free living stage. As an example, nostoc, Trebuchia, even they not produce the lichen, they are present in the free living stage. So these are some of the images of these photobionts. Now this is the algae Trebuchia. It is a round shaped organism, and you know this is the cyanobacteria. It is a nostoc, and it has the this chain like structures present in their body. So these all are taken under the light microscope. And this is actually very interesting literature very recently published. Now, here they mention how this lichen form with the combination of different fungal species and the photobiont species. Now, if I go to this first image, now you can see this F1, F2 mean different fungal species, but here they have taken two genera, but their species are different. And after that, this P mean the photobiont, and how they combine into form different species in this stage. And in another stage, you can see there are three type of species of fungus, and then this three type of fungus incorporate with one type of algal partner, and after that you can see, although there is a single algal partner and there are different type of fungal partners, and finally they produce three type of species here. And this is another one where only one fungal species there, but different photobionts are there. And in the combination of these P2 and P3, these are two types of photobionts. When they combine with the single mycobiont, now they again form a same species. And this is another explanation where you can see one type of fungal species, and there is a two type of photobiont species. Now this is P5 mentioning about the green algae or the cyanobacteria, and this is normal green algae. Now, although there is a single type of fungal species there, and they found two types of species here. So now this is this is the one recently published by the scientist with the laboratory evidences. So with that, I think you can get a thorough idea about how this lichen form with the help of different fungal species as well as the algal species. And this is another one where they explain how this lichenization process happening. This also published in 2022. Now, you know, when fungus and algal combine together and then they form lichen thallus, And what would happen in the relikenization process? At this stage, they separate from the main lichen thallus as the algae and fungus, and after that they recombine, and again they produce their lichen thallus. And this is another phenomena. Actually, it is completely different from this one. This is they call it as a translichenization, where a lichen is there and another fungus just landed on the lichen thallus, and after that fungal species just germinate on the surface, and after that they penetrate through the lichen thallus, and what they do is they get the advantage from the photobiont, and then what they form another type of different species just on the surface of the lichen. So we call this type of lichenicolous lichen, because there are some lichens even just on the top of the lichen you can see another type of lichen. So this is how they form another lichen thallus on the same lichen thallus. So this is very interesting article. I mean, if you have time, just please try to read this one. You can get more novel knowledge related to the this formation of lichen in this article. So basically, I am going to discuss about in this talk how we can identify lichen based on their morphological characteristics, then anatomical characteristics, and their chemical characteristics. So now at the beginning I mentioned that if you can distinguish lichen from the other organism, so that would be the first step to identify the lichen. And after that, actually, we have to go through these different morphological, then anatomical and chemical characteristics. Then only you can identify it up to a genus or species level. Actually, apart from these three characteristics, we have to go for the molecular biology or the genetic level also, but I am not going to discuss about that here. So using these the four areas, you can finally identify a lichen up to a genus level, or most of the time you can go to the species level. So let's first discuss about the morphological characteristics. I mean, that means when you take a lichen into your hand, onto your hand, or if it is available in the natural environment, with the help of hand lens, actually you can see many of the characteristics that way I am going to explain as a morphological characteristics. But for the anatomical characteristics, always you have to use microscope or either light microscope or stereo microscope. Without such facilities, it's really difficult to identify these anatomical characteristics. For the chemical characteristics, actually you have to do some analysis like thin layer chromatography or HPLC analysis for the identification of lichen. Now, considering the thallus morphology, now you know there are basically four types of lichens available. Now you remember, now you see the lichen. Now first you identify, okay, it is a lichen that is fully differentiate from the other mosses or other cryptogamic species. Then you have to come to some idea whether it is a fruticose lichen, foliose lichen, or crustose or scamulose lichen. So with the having that knowledge, then you can go to the second level. So where we can categorize all those lichens into four mobs. One is a fruticose lichen. Those are very easy to identify because they are very large in size, and always they have attach to their host species using a we call hold fast. That means sometimes they just hang on the lichen, or sometimes they are in an upright position. So, and other characteristic is that that lichen you can very easily separate from the host, or where that lichen attach with the other host species, either rock or either bark or whatever. And also most of the time they are, I mean, if you take a if you closely look the lichen, you can see most of them are either a round shape or they are strap shaped. I mean, you can't see properly differentiate upper or the bottom level of the upper part of the lichen thallus, like the next one I am going to explain. So the footicose is the one type of lichen, and the other one is the foliose. So you see they are flattened branching lobes, and they also very loosely attached to the substratum. Most of the time they are leaf-likes. That's why we call them as the foliose. And also, not like in the fruticose lichen, they have upper and lower surfaces. Now, if you turn it upside down, you can see the completely different lower and the upper surface. So that would be another character that you can identify it as a foliose lichen. Then the other one is the crustose lichen. Now, this is completely different from the previous one. That means foliose lichen or the crustose lichen. Why is that? They are fully flattened, and some are scale-like, and they don't have lower surface. Why is that? Now, if you want to see the lower surface, actually, it's really difficult to separate them from its host. So, whenever you want to take it out, you have to take the its substrate as well. Like, as an example, if it is on a rock surface, you have to cut the rock or break the rock. Then you have to take it with the piece of rock. Otherwise, it's really difficult to take it out from the substrate. So those type of lichens we call crustose lichen. And there is another one we call scamulose lichen. Actually, it is an intermediate type of lichen in between foliose and crustose lichen. And they have, specifically scale-like slopes present, very tiny scale-like slopes present, and they are very smaller than the foliose lichen. So, depending on the definition, some scientists define some other lichen types as well apart from these four types. But basically, we can identify these four types as scamulose, crustose, foliose, and fruticose. Now, you remember, now you found a lichen, you separate it from other species, cryptogamic species. Then now, when you look at it, you can have some idea whether it is a foliose lichen, crustose lichen, or any type belongs into these four types. So then that is the second step. So then what you have to do is, if you want to identify further, you have to have some knowledge about what are the other characters you can observe on the surface of the lichen. So let's discuss some of the main characteristics of the lichens. Then only having that knowledge, you can go through different lichen keys. Those are available in the internet. There are many lichen keys available. So when you go through those lichen keys, you can at least you can go up to a genus level. You have to have that basic knowledge. Without that knowledge, it's really difficult to go through the identification keys. So let's look at some of the common structures you can find on the lichen thallus. Now, One structure is we call apothecium. Now here I am not going to discuss further because I will later discuss this structure. And apart from apothecium, you can see these terms also very important, right? It is, I mean, you have to remember those terms because in the taxonomical purposes, you always have to use those terms for the identification purposes. Now, one thing is we call Soredia, and the other one is we call Isidia. Now, actually, if you closely look at on the top surface of the lichen, sometimes you can see like powdery masses or powder-like masses coming out of the lichen surface. So those are we call Soredia. Now you see, if you take a cross section of the lichen, here you can see now this component actually you can see in the powder form. Here you can see them on the lichen thallus. Actually, these are these Soredia and Isidia are kind of vegetative reproductive structures. Why is that? Now you see this this powder particle composed of that algal component and the fungal component. So what could happen is that when due to some external factors like wind or animal or any other factors or any water or whatever, they spread around the environment, and as it already available it component this algal and fungal partner, what they can do is they can start the germination of lichen thallus in the new place. Even in this Isidia also, you can see this finger-like projections. When you observe them under the most of the time under the light microscope or your hand lens, you can see them very easily. So that finger-like structure also consists this algal component and the fungi component. So what would happen due to external forces, these structures very easily detach from this thallus structure, main thallus, so that this component go away from the main thallus, and after that this finger-like structure can regenerate its thallus. So it is they usually use for their vegetative reproduction, but these are very, very important structures we can use for the identification of lichens. Now, when you go through a key in some section, it says the lichen with the Soredia, then without Soredia, or lichen with the Isidia, without Isidia. Likewise, when you go through the dichotomous key, it's really important to know whether any lichen consists either Soredia or Isidia. So having that knowledge, you can further go in a specific key, then you can use that character for the identify the lichen. So now when compare with the Soredia and Isidia, you can see the Soredia is not covered with the epidermis of this lichen, right? But here you can see this Isidia is covered with this actually the outermost layer of the lichen, so that you can see this is a powder form, but here you can't see it as a powder form, but you can see it as a kind of finger-like structures projecting from a lichen thallus. So that is one of the basic structure that you need to understand for the identification of lichen. And the other one is the rhizines. Rhizines means like root system, but these are not actual root system. These are just outgrowth of a fungal partner, but they are you can see at the lower surface of the lichen where they use them to attach to the substrate. And also sometimes they excrete some chemicals to the substrate, or sometimes they absorb something from the its substrate. Now, as an example, so far scientists believe that if lichen present on a stem of a tree, there is nothing happen with the tree species. They believe that it is just the place to survive the lichen. But later on, very recently, scientists found that actually what could happen, they penetrate their rhizines through xylem and phloem, and they excrete some chemicals for the plant species. So now we know that not all the lichen, right? But some lichens, when they present on the surface, especially in the bark, they penetrate to the xylem and phloem. However, many lichens, those are present on the rock surfaces, they excrete some chemicals. So those chemicals are, it is, I mean, we can discuss in different ways, right? One is that if they are, just imagine if they are present on the normal rock surface, so those chemicals are very important for the rock weathering. So that because of, the plant succession, lichens are the pioneers organism, they invade that bare land. After that, they excrete some chemicals to the boulders or the rock surfaces, and they helps to digest rock particles, and they helps to formation of the soil. But just imagine if they are present on a monument. We know that monuments are very important. Now, when they are present on the monument, even in that surface also, they do the same thing. Because of the excretion of some chemical, it helps to digest the surface of the monument. Many of the European countries having that issue actually, rather than other acids, even lichen also excrete some chemicals. So those are, I mean, helps to degrade. So it is a kind of disadvantage. So anyway, now this is another character where you can see a different arrangement of the rhizines. They are now this is the actual photographs of the rhizine, how they looks like. So this branching pattern also we use for the identification purpose. There are some rhizines available. Those are branched dichotomously. So that dichotomously branching character we can use for the identification. In some cases, you see here, you can't see any branching pattern. So it is another character of that specific lichen. Now here it is another way, it bush-like, so that it is another character. So likewise, you see this type of rhizine pattern also very important for the identification of lichen. And the other one we call cilia. Now cilia is actually one mislead with the rhizines and cilia as well, because cilia always present at the edge of the lichen thallus, right? But rhizines, they most of the time they not present at the cilia, at the edge of the lichen thallus, but having the experience. When you observe them under the microscope, you can easily identify whether it is a cilia or rhizine. So presence of cilia or absence of cilia is another character where we can use this character for the identification of lichen. So remember that rhizines or cilia also very important. And also, as we discussed in the previous case, Soredia and the Isidia. And apart from that, you can use some other characters for the identification of lichens. Now, one example is that a pycnidia, right? You can see on the top of the lichen thallus, you can see tiny dots present, right? Now, these are actually we call lichenicolous fungus as well, because there are some lichen species are there, they are very specific for some other fungal species to grow on the thallus. So you know, pycnidia is a or pycnidiospores, you know, it is a asexual spores. So if you take a cross section of this one and if you observe them under the light microscope, you can see how it looks like. Now I will come to later. Now even in the lichen produce their sexual fruiting body we call perithecium. So perithecium also sometimes looks like a pycnidium. The only difference is that in the perithecium they produce ascus inside the perithecium, but here you can't see any ascus inside the pycnidium, and even inside a single pycnidium most of the time they produce sometimes millions of spores. So large number of spores you can see inside this pycnidium. So this the presence of pycnidium also not it always use, but sometimes we can use that character also for the identification purposes. And the other character is cephalodia. Cephalodia again you see when you look at the lichen thallus, you can again see some type of blackish dots on the surface of the lichen, but those are not the pycnidia as we discussed in the previous slide. If you closely observe them, you can see it is a kind of outgrowth from the surface of the lichen, where it consists a large number of photobiont cells with the other algal fungus component. So, right. So this also a kind of vegetative reproductive structure. So what could happen? So this structure can detach from this area, and they can easily go to another new environment, and from that environment they can regenerate the new thallus. So likewise, I mean now we are discussing what type of structures present on the thallus. Now we discuss several things. So cephalodia is another structure you can identify on the thallus of the lichen, but you know it is I mean cephalodia are not common as pycnidia, but they also another type of blackish structure. Sometimes you will mislead with the pycnidium. And also, I mean, apart from the main thallus, if you observe any lichen, sometimes just below the main thallus you can see another thallus where it extended further at the edge of the main lichen thallus. So that type of thallus actually we call prothallus. So whether the presence of prothallus or absence of prothallus also another character of the lichen identification. So this is very important because many of the lichens species with green algae as the photobiont, those type of lichen most of the time they produce prothallus. So we call this the below thallus as the prothallus. You can see this blackish one, it is a prothallus. And also in some lichens actually you can see two type of thallus. Now one thing is the erect one, and the other one that is parallel to the surface. Especially this type of thallus you can identify with the Cladonia species, and this you know these are their footing bodies, and even footing bodies also they contain some of the scale-like structures. Now this is another type of scamulose thallus where you can see two types of erect thallus and the other one that is grow parallel to the surface. So likewise you can see different patterns of the lichen thallus. Okay, and those are actually most of the time vegetative reproductive structures now so far we discussed. And apart from that, the major component of the lichen is they are sexual structures, either apothecium or perithecium. Now I will show you some images of apothecium and perithecium, but before that, just try to understand what is the apothecium and perithecium. Now, if you take a cross section of the apothecium, right? Now you can see it is like a cup, and inside the cup you can see the ascus, and inside the ascus you can see the ascus pores. Now this perithecium also another extension of this apothecium. Why is that? Now just imagine this either wall of this hymenium, they grow like a cup like this, and after that what would happen, it make a very small opening there, and here it is a big opening, but here it is just a small opening we call apothecium. So it looks like a it's, earlier I mentioned pycnidium, so it looks like a pycnidium. If you look at from the top of the lichen, you can see it just a opening, or the small opening you can see, but the main part actually present just below the opening. So even this perithecium also, you see their ascus are arranged like this, and inside the ascus you can see ascus pores. So I'll come to later how this ascus arrangement and how these pores are vary from one species to another species. So you have to thorough knowledge about how looks like apothecium, how looks like perithecium. Now you see these all are different types of apothecium, right? Now this apothecium, you can see the margin of the apothecium, and this is just a substrate in each places, and inside this or the middle part of the apothecia, where you can see this opening of the ascus, right? Now when they become mature, what could happen, they produce ascus, and ascus pores come to the top level, and after that, due to any external factor, they spread throughout the environment. Now considering this ascus, now you know, now you identify or you found a lichen, and you identify whether it is belongs to that 4 type of species. Then you, now what you are doing, you are observing these different characteristics, right? Now you check whether it has a Soradia or Isidia, whether it has rhizines, and how rhizines looks like, and then you observe some other characteristics, any black dots available or not, like that, or whether it is it has cilia or not. Then you come to the sexual identification of the sexual structures, where as I mentioned, there are 2 types, apothecium and perithecium. Now these are the apothecium, right? It is a cup-like structure. Now when you look at the top layer, now they looks like a round shape. Now when you look at apothecia, right, basically you have to pay your attention for 3 characteristics of apothecia, or there are 3 types of apothecia available. One apothecia we call lecanorrhine, and the other apothecia we call lecidine, and the other one we call bioterrine. And apart from these 3, there is another type we call lyarellate. I will come to that one later. Now, so what are the main 3 differences between these 3 types? Now, when you look at the lecanorrhine, you see this apothecium margin always same color as its thallus, right? Just pay your attention to the margin of this apothecia. In every cases, you see this apothecial margin and the thallus color are same, right? So this type of apothecia we call lecanorrhine apothecia. In each of these cases, you can see this apothecial margin is color always similar to the thallus color. And when it comes to this one, right, lecidine type, although they have their margin or the apothecial margin, but this apothecial margin color is totally different from the thallus color, and also that margin color is almost same as the inner part of the apothecium, right? So most of the time this lecidine apothecia are black in color, so that you can easily identify them. Now I think you can get idea how they distinguish from each other, right? Now here blackish margin is there, and it is the margin color is different from the thallus, and also the margin and the middle part are same in color. But when you come to this one, right, bioterrine, it is totally different from these two types. Now when you look at the margin, always you pay your attention to the margin color. So this margin color is totally different from the thallus color, right? You see, and also here this margin and the middle part of the apothecia also a different color, right? So those are the main three types where you can identify whether it is a lecanorrhine apothecia, lecidine apothecia, or bioterrine apothecia. So now when you look at the lichen, right, now you can get some idea whether okay, it has a apothecia, then what type of apothecia is there, whether lecanorrhine apothecia, lecidine apothecia, bioterrine apothecia, because these characteristics are very important when you go through a lichen identification key, because in some point in the key mentioned that whether that lichen, okay, it mentioned it has apothecia, then perithecia, then you go through the apothecia part of the apothecia, then after that it further divided into whether apothecia is a lecanorrhine, lecidine, or bioterrine. Then you have to select the appropriate apothecial type, then only you can go for the further steps, right? So likewise, when you see the apothecia, you have to identify whether it belongs to these three types. And also there is another one we call, other one is the perithecia. Now you see perithecia, how it looks like. Now you can see just the opening of the thallus, right? Now there are different types of perithecia available. In one case, their walls are carbonized. Carbonized mean black in color, and if you want to take a cross section, it's sometimes really difficult to get the cross section, right? Now if you open this perithecia, they looks like this, right? And this is another one, but this has not fully carbonized perithecia, and even if you take a cross section of this one, you can see this ascus inside the perithecia. Now actually you see they are most of the time embedded on the bark or the lichen surface. You know, lichen present most of the time present on the plant stems or the bark, so they are just embedded inside the bark. So what you have to do is just observing the dissecting microscope, you have to take a cross sections like this, and after that you have to identify or you have to observe under the microscope. Even in this one also, in the lichen case, they have mentioned whether it has a perithecia, then they divide whether perithecia has the carbonized involucrum or non-carbonized involucrum, right? So if you have that knowledge, then you can go further on this key, right? Sorry, actually this has to come earlier than the previous one, right? Now you know there are three types. I discussed three types of apothecia: lecidine, then bioterrine, and lecanorrhine. So this is another apothecial type we call lyrellate apothecia, right? So they are very elongated apothecia, right? Now they can you can easily identify. You see, I mean, different arrangements are there, right? Different arrangement are there. So now basically this type of lyrellate apothecia present in the family Graphidaceae, right? So we have different types of Graphidaceae keys. So what you have to do is whenever you found this type of lyrellate lichen with lyrellate apothecia, you have to refer the any Graphidaceae key. Then they mention so many characteristics, right? I'm not going to explain all the things, but at this level, at least just try to understand whether it is a lyrellate apothecia or not. That enough, right? Now, if you want to further observe, what you have to do is you have to take the cross section of this lyrellate apothecia. You can take in any place of this apothecia. After that, you have to observe them under the microscope. So then only you can get idea how they looks like, right? Okay, so far we discussed about the morphological characters, right? That means once you found a lichen, you have to pay your attention to identify the characters or the other structures. Those are present on the surface of the lichen. So having that knowledge, you can easily, understand, or you can go at least halfway of a lichen key for the identification purpose, right? But apart from that, actually we need to know what are the anatomical characters of lichen as well. So yes.
Okay, ladies and gentlemen, if you have any questions, please raise your hand. I will allow you to talk then. Sheetal, yeah, please.
Good afternoon, sir.
Yeah, good afternoon.
So my question is, when you told us about the process of translichenization, you said that the new species arises from the original one. So
Yes.
that new species, is it the same one, or obviously or a different one?
Yeah, most if it is a new species, actually it is their characteristics are totally different from the previous one. So actually sometimes when you observe the anatomical characters, sometimes you can't see proper difference, but if you go to the molecular level, then only you can get idea whether it is the new one or not. So I mean, without having molecular data, sometimes it's really difficult to say whether it is a new lichen or not, especially if morphologically if they are same in morphologically, then it's really difficult. So that what we have to do is always we have to extract the total genome from the fungus. Usually we use ITS. Then I mean, somebody argue that now it is a combination of fungal part and mycobiome, but that ITS one always react with the fungal component, so that we no need to worry about the algal component. So that we can go for the sequencing, PCR and then sequencing, then only we can come to a conclusion that after phylogenetic analysis whether it is a new or not.
Okay, so can we say that the process of speciation is
Yes.
more faster in lichens?
I think we can't say, because there are many lichen species not so far identified, right?
Okay.
So that nobody has a thorough idea how long it will take to differentiate into new species or not. But as many of the areas are not properly investigated, in that sense, whenever you observe any unexplored area, there is a high chance to find a new species.
Okay, sir. Thank you. Sir, I have another question, but I think there are more people are waiting for this, so I'll ask it later. Thank you,
Yeah.
sir. Thank you for your
Okay.
answer.
Okay. Okay.
Nikhil Verma, you can ask your question, please. Okay, anybody has any other question? You can raise your hand and speak. Okay, Madam Shashireka.
Good morning. I'm sorry I joined late, but I had a very simple question. There's this lichenization which happens between the algal and the photoband, sorry, algal
Yes.
and the mycoband. Is it possible that in the natural situation, there are so many other fungal spores that
Yes.
are present there? And if
Yeah.
probably algal also must be present, is it
Yes.
a confusing situation for the fungus to find the partner?
Yes, madam. Actually, there are, considering the lichen thallus, actually, we can identify three types of fungal species. One is lichenicolous fungi. That means another fungus that present on the lichen surface, but they are not involved for the formation of lichen. And the
Okay.
other one is lichen forming fungi. That means that is the fungus they actually involve for the formation of lichen.
Yeah, I
And the
understand.
other one is endolichenic fungi. That means another kind of folliculus. That mean lichenicolous lichen. That mean now in that situation they are present inside the lichen, but there is no any interaction between the lichenized fungus. They just uh, uh, live there. I mean, they use that lichen as a host species.
Yeah.
So that is the three situation. That only thing is that now in the laboratory level, it is really difficult to separate or the separate that lichen forming fungi. Other two species are grow very well, but it's really difficult to separate that. I mean, lichen forming fungi. So
Yeah.
that is the situation. The only thing is that now when you want to observe that lichen forming fungi, usually we sterilize the surface of the lichen, so that what would
Okay.
happen, we can remove all the other
Other type of soaps.
lichenicolous fungi. And if the lichen contain, as I mentioned, this apothecia or perithecia, so you have to take the section, and there is a method we call single spore method. So
Yeah. And
using that method, you can separate the lichen forming fungi.
so that means it is from within the thallus.
Yes, within the thallus.
Okay.
Yes.
Okay. That has always been foxing me, I
Yeah,
should say. I
that
used to
is
wonder.
the issue actually come with many people. It's really distinguish which one is the lichen forming, which one is the endolichenic, and which one is the lichenicolous fungi. So
Right.
at least we can remove that lichenicolous one if you sterilize the surface of the lichen before you separate the lichen forming fungi.
Then what about the algal partner?
You mean
Algae,
uh,
photoband.
yeah, photobind actually most of the situation it is same, right?
Yeah.
Yeah,
Okay.
it is. I mean, there is no much involvement, but different photobinds are in different species. Now, as an example, if you take Trebuxia, that is the most common algal species, and apart from that, there is another one we call Trentifolia. So
That is a
orange
sign.
color, yeah,
Oh,
yeah. So that one we easily distinguish because if you scratch the surface of the lichen, sometimes you can see somewhat orange color.
okay.
Then definitely it is the Trentifolia. But if
Okay.
it is in a pure green in color, then it is the Trebuxia. I mean, apart from those two, some green algae are there, but those are the common species you can identify in the lichen.
Blue-green is also involved in this, right?
Sorry?
Blue-green algae, cyanophycean
Yeah,
members.
so cyanobacterial one, actually you can easily distinguish when you look at the lichen thallus. That means many of the time those lichens are darker in color, not
Okay.
greenish or gray color.
Okay.
So whenever it is a darker in color, like dark grayish or blackish color, then most of the time they are the fungal algal partner is the photobiont. So
Okay.
that also very most of the time anabina or nostoc is there, so they can we can observe them very easily under the light microscope.
Okay.
Yeah.
Just one last thing.
Yeah.
I have been observing in the forest that some of the trees are covered with white patches. White patches and people, whether it is lichen
actually,
or
sometimes madam, we have to mislead with some other fungal species as well. Now there are some lichens, actually I have not mentioned here. There are some lichens, those are a sterile situation. They never form any footing bodies. Now in that sense, the main thing is that we have to scratch the surface, and then you have to observe under the microscope whether it is whether lichen species contain any photobiont. Then only we can say it is a lichen or not. So if it is not, then most probably it is
it could be
another type
fungus,
of fungus. Yes.
which could be parasitic also, right?
Yeah. Yes.
Yeah. Thank you so much. Thank you so
Okay,
much.
welcome.
Dr. Shankar Bhatt, do you have any question?
Yes. Sir, I am a bit curious to know, are there any parasitic lichens?
Yeah, now as I think I mentioned that now recently some scientists found some parasitic lichen. Many of them are not parasites, but there are some lichen species. What could happen is that they penetrate their rhizines into the xylem and the phloem. After that, they excrete some chemicals. Still, they don't know what is the purpose of excreting chemicals. But in that situation, scientists found that there are color deformation and some other characteristics in the plant species. Yeah.
Further, are there any infection from other fungi on lichens?
Yes, there are many. As I mentioned, there are some lichenicolous fungi. So they are actually parasites for the lichen. They just land their spores on the lichen thallus. After that, they just grow on the lichen and they absorb everything from the lichen. So that why is that the photobiont is there, so it produces starch and everything, so that the newly came lichen act as a parasites on the lichen thallus. Actually, it is nowadays emerging area where people are working on lichenicolous fungus because they produce again many antibiological or the antibacterial, antifungal chemicals. So that, I mean, in that sense, they are very important. But if you consider about the lichen, it is not good for the lichen thallus.
Thank you, sir. Thank you very much.
Okay.
Mr. Tejas Kumar. Mr.
Sir,
Tejas.
yes, sir. Sir, I would like to know what is the effect of rainfall or the moisture conditions on the diversity of lichens. Will it increase the diversity, or in any moisture condition are the diversity more, or in dry conditions the diversity more?
No. The actually rainfall also another factor. Rather than rainfall, humidity or the moisture content is the important factor. But if it is a rainy area, but they like if the lichens are in a shady environment, then actually you can't see high diversity. So, but if another area is there, that area is fully open, and that plants can get enough sunlight and again have high humidity in that areas, most of the time you can see a large number of lichens as well as the high diversity. But it is obvious that in dry areas you can't see that much of diversity in lichens.
Sir, what about in forest areas where there will be forest canopy and the light cannot enter that much?
Yeah.
In
Now,
forest areas, will it be more or what?
yeah, thing is that actually now in the forested areas, we basically focus on the lichens present on the bark of the stem. No, so where we can reach up to a certain level, then only we can study them. But we don't know actually at the canopy level how many lichens are present. So it depends actually. In the canopy level, they get enough sunlight, and that the fungus or the photobiont can fully photosynthesis, and therefore most of the time at the canopy level you can see a proper lichen community rather than the bottom level. The thing is that if it is a forest, actually if it is possible, we have to survey, or we have to observe the different level of the trees, then only you can get some idea. But I am 100% sure at the bottom level you can't see that much of diversity because of the sunlight, less availability of the sunlight.
Thank you, sir. Thank you.
Okay.
Miss Madhavi.
Hello, sir. Very good afternoon. Sir, my question is like among different genera of lichens, are there any genera which produces dormant structures to avoid the adverse conditions, sir?
You mean dormant structure?
Yeah, are there any lichen genus like which produce dormant structures to avoid the adverse conditions, sir?
Yeah, I mean, that vegetative reproductive structures sometimes act as a dormant structures. As an example, this Isidia, because it has a cover there, so that they can survive with the harsh environmental condition. But actually, there are no more structures available now, like in other organisms. But that cephalodia is another one. It can survive up to a certain level. However, that if water or if less water and high temperature, whatever, what would happen, that lichen thallus can shrink, right? Then what would happen, lichen photo always all the activities depends on the photobiont, activity of the photobiont. So that what would happen, that lichen the mycobiont part act as a protective structure for the photobiont, so that during the dry season they shrink and they protect the photobiont. Now sometimes it is just a mechanism, but they don't have such type of structures in most of the cases. Using that mechanism, they survive under harsh conditions. Now once they get the enough rain, then what would happen, that water absorbed by the fungus, and again that photobiont reactivate for the photosynthesis process. So that is a kind of mechanism you can observe in the lichen. Right. I think so far we have discussed about the morphological characteristics. Now then we have to go for the anatomical characteristics, or we call basically we pay our attention to observe the internal structures. So that also very important, because without having that knowledge, I mean, you can't identify sometimes at least up to the genus level, because this anatomical characteristics play a major role for the identification of lichens. So let's look at what are the basic anatomical characteristics that we have to pay for the identification of lichens. Right? Now, considering the lichens, if you take a cross section, it contains various zones or region of the fungal tissue, and also in most of the autotrophic symbionts restrict to a definite layer. But there are some lichen actually, if you take a cross section, you can't see any confined layer in the photobiont layer in the lichen thallus. So let's look at if you take a cross section, how look lichens. Now this is one of the electron microscope image. Now you see basically at the top layer you can see the cortex, right? Although we call it as a cortex, most of the time it is not a kind of a barrier like cuticle of the plant, because in any heavy metal, any gases or whatever, they just penetrate through this lichen thallus. So that's why most of the time we use lichen as a good bioindicator of the environmental pollution. There are many studies going on, many scientists looking for heavy availability of heavy metals in the lichen thallus. So once getting the idea about the availability of the heavy metals available in the lichen thallus, you can get some idea how far polluted in the environment and so on, right? So however, there is a upper layer we call the cortex, and just below the cortex you can see the medulla. Either cortex or medulla, the major part is the fungal component, but in some lichens actually, now you can see in this image, they are confined to a layer, right? And that layer always close to the upper level of the lichen thallus, because if they are present at that level, so they can get enough sunlight. So you can see these are the photobiont, and they are just embedded in the lichen thallus. And in some cases, I'll show you another picture. Now this is the photobiont. It is a blue-green algae, right? So most of the blue-green algal lichens, if you take a cross section, you can't see this type of confined layer. They are present just throughout the lichen thallus, right? Now, as you can see in this one, this is another type I mentioned at the earlier. In here you can see the confined algal layer, but here you can't see that type of algal layer. They are, in every places you can see, right? So that also another character actually you can get for the identification purpose. And also this upper cortex and lower cortex, that also another good character, because in some lichens you can see this either part, that mean upper cortex as well as the lower cortex. There are some lichens, they don't have lower cortex, although they are foliose lichen. You know, the crustose lichen, obvious that they don't have the lower cortex, but in some foliose lichen also they have lower. Some lichens they don't have lower cortex. So that also another character for the identification purpose. Now you see, if you take the cross section, how the arrangement of the photobiont and the other part of the mycobiont in the lichen thallus. That structural arrangement is common for their fruiting bodies as well, right? Now, if you take a fruiting body, now you can see this cortex layer is continuously go through the apothecium as well, right? And inside the apothecium, you can see this fungal component, and once they get mature, they form this type of apothecia, and finally they produce their ascus and the spores. Right now, that the availability of number of layers also important, and that we can observe under the only under the microscope, otherwise you can't observe that. And similarly, once they mature, they form ascus, right? So the arrangement of spores inside the ascus also very important in most of the cases, because it depends on the species of the fungal partner, right? Now you can see in this ascus, we know usually inside the ascus they produce only 8 spores, no? But in some cases they produce more than that, right? So this is one example, and you see as there are a large number of spores, they are in different layers, right? There you can see different layers you can observe, and this is another. This is the most common one actually. You can see they are in a single layer, and those 8 spores are stacked in a single line. And this is another situation. I mean, these are just an example, right? There are many arrangements are there. Now here, all the spores are arranged in 2 levels, right? Or the 2 lines. In one side 4 spores and the other side 4 spores. And here it is a completely different one. They produce ascus, and large number of ascus spores you can see. And also, actually, I have not included that character here. Even this shape of the ascus and the tip of the ascus also, the shape of the tip of the ascus also, most of the time used for the identification purposes, right? Now and I forgot to mention one thing. Now, whenever you observe under the microscope, actually we have to add some color reagent like cotton blue or something like that. So then you can observe them very clearly. Then, I mean, as an example, if you want to observe what is the shape at the apex of this ascus, then we have to color them, right? Or the stain them. Then that structural arrangement, structural shapes, and everything you can observe very clearly. So that means this is another anatomical character. Then you have to understand what is the shape of the ascus. Now you see this is one of the elongated one, and this one is the some of globose one. And also I'll come to that one later. How spores looks like, right? That also very important. Not only for the lichen, even in the other identification of the fungus also, we always consider about those characters. So in the lichen also, we have to observe such characters. I mean, without having such knowledge, again, it's really difficult to identify the lichen, right? Even this sometimes this Hymenium character also sometimes scientists use, whether it is a branch one or the single one. Likewise, they consider about that character as well. So then pore shapes and their septation also very important, right? So these are some of the spore types we can observe in the lichen species, right? Now you can see they are round shape or oval shape. Some are elongated one, right? Some are spindle-like, right? And also the number of septation, right? Now here you can see in this spores, you can't see any septums or septa inside the spores. But here you can see the large number of septums arranged inside the spores. Now here single septum is there, and this has several septums, right? Likewise, and this also several septums. So likewise, you have to understand how spore looks like, and whether it is elongated or round one, or likewise you have to consider about the shape. And sometimes here I have not mentioned it, whether it is a color one or not, right? You know, many spores are hyaline or the colorless, but in some species they produce colored spores. So that colored spores is a prominent character for the identification purpose. Now, if you go through a dichotomous key in one place, sometimes it mentioned as it has a one septate or one septum spore. Then as the next step, it mentioned whether it is a brown color or hyaline, right? Then you have to properly identify whether it is most of the time you can see brown colored one, whether brown one or the hyaline one or the colorless, right? So that also very important. Now I think, and I forgot to mention, sometimes some spores produce some sheet-like structure, right? Now there is a genera we call Megalospora, right? Now in the spores, sometimes they produce sheath, and also sometimes they produce thin or thicker spore wall as well, right? So that character also very important for the identification purpose. And this is another set of spores, especially we call this type of spores muriform, right? That means in this spores you can see they are crosswise and longitudinal way. They break into small component. That means large number of septums present in the either directions, right? So those are we call muriform. Now you remember I mentioned about that lyrellate graphidaceae species, right? Many of those species you can find this type of muriform spores, right? They are very beautiful when you observe under the microscope. And sorry, I forgot another one. You have to mention about the size as well, right? Because that also sometimes species wise vary. Now, as an example, if you take these two, or let's say this two type, now you see this also muriform, this also muriform, but although this is the muriform one, spore size is smaller than this one, right? So it is completely a different species, right? So that spore size also very important. Now, when you want to give the spore size, you have to give the length as well as the width. And if you want to give a medium value, then mean value, then what you have to use, at least you have to get the length and the width of at least 10 spores, right? Then only you can get the final value. After getting the mean value, you can say about the exact size of the spores. Because in the same species also you can see little bit variations in their length and the width, so that when you give a mean value, so that is most scientific, right? And this also another type of spore. Now when you observe them under the microscope, you can see this type of different shape of the locules you can observe, right? So those are sometimes unique for some genera. Like there are some perithecium producing species are there, Porina, and I can't remember some of the name. Anyhow, most of the crustose species, they produce this type of spores, and also sometimes some heterodermia, there's a genus heterodermia, they also produce this type of spores. So now you see, when you go to the anatomical level or the microscopic level, large number of variations you can see. Actually, most of the time, this spore characteristics are very, very important rather than other characters, because some species, I mean, sometimes they appear in the same way, but if you go to the spore characteristics, sometimes that spore characteristics are totally different from one species to another species. So that it is very important to observe spore characteristics. That the point is that sometimes some lichens don't form apothecia or perithecia, right? So in that sense, it bit difficult to identify some characteristics. Actually, in that sense, what we have to do is we have to go for the molecular level. Otherwise, it is really difficult to identify such lichen species. So if I describe about their reproduction, you know, they usually do their sexual reproduction with the help of ascospores, right? And also they do some their asexual production, like I mentioned, mainly on the fragmentation of the thallus or fragmentation of the isidia, or sometimes formation of the soredia and so on. So their asexual reproduction is more prominent, as I shown at the initial at the beginning. You see how this soredia form, right? Those are just powdery like structures, right? They detach from the main thallus, and after that they form a new thallus. And isidia also some of the important characteristics, and there are some other minor characters also. They are pseudocyphellae and cyphellae I have not mentioned, but if somebody needs more information, please just contact me so that I can help you for the identification purpose. And many times people ask, can't we produce lichen thallus inside the laboratory, right? Now, people believe that if you separate their fungal partner and the mycobiome partner, after that you recombine them, whether there is a formation of lichen thallus or not. So far, nobody able to make a natural, I mean, thallus like it is in the natural environment, right? But they come to a certain level that they form some lichen thallus. Now, only very important thing is that although they don't form a proper lichen thallus, the secondary metabolites are always similar as the original one. So that is a good sign that you know, lichen show very slow growth rate. Now you can see here 1 to 2 millimeter per year, so that it takes long time. And if you want to take them for your research purposes, then you know sometimes it is not good practice because of their slow growth. Sometimes we vanish some certain species from their natural environment, so that whenever you found an interesting thing, it is better to resynthesize those chemicals inside the laboratory, right? So therefore, scientists recently completed this one, Now you see one of the good example. They separated their photobiont and mycobiont, and after that they combined together. And you see this is they are resynthesized thallus, but this is the actual thallus. When they are present in the natural environment, it looks like this, but you see it is totally different from the original thallus. However, fortunately, as I mentioned earlier, now the secondary metabolites present in this resynthesized lichen and the naturally available lichen are 100% same. So that it is, I mean, positive sign that if somebody wants to study on the secondary metabolites available in this original species, what you can do is you can resynthesize them inside the laboratory, and after that you can extract such chemicals for further studies. So that is a good signs of synthesis of lichens in the laboratory, right? So those are the main anatomical characters that we have to use for the identification purpose. Then we have to go for the chemical characteristics. So I will go a bit fast through this one. You know, lichen produce secondary metabolites, and those are, I mean, unique for the lichen species, right? That means such secondary metabolites not produced in other organisms, right? Therefore, the lichen secondary metabolites are very, very important. Now, considering the lichen thallus, up to 2019, scientists have found more than 1,000 secondary metabolites from the lichen, and they are present in their medulla, their cortex, and their ascus and prothallus and everywhere, right? The main purpose of having the secondary metabolites in the lichen is for the protection from high temperature and the UV, right? And also excess water and the drought. And also sometimes they can protect from their anti-herbivores and microbes, all right? Because of that reason, they produce such secondary metabolites. Now when you look at these images, you see they are very colorful, right? So these colorful characteristics are generated with the help of secondary metabolites, right? Now when you extract the secondary metabolites from these lichens, you can see so many lichen compounds, some are very unique for some lichens, right? So, and also because those secondary metabolites having so many antimicrobial activity, nowadays scientists are heavily work on that, right? So in the scientific manner it is okay, but if you consider the lichen protection and so on, sometimes it is not so good, because some are trying to take the lichen from their natural environment, so that it's really difficult to make such lichens in that their natural environment, right? There is a good example in India. There was a lichen in Himalaya Pradesh, as I remember, and what has happened, the people harvested that lichen for different purposes, because they can sell them out very easily. And later on, after some decades, the scientists found that there is a one lichen Parmelia species, I can't remember the species name, so that is already extincted from the environment, right? Because of the heavy harvesting of the lichen thallus. So we have to pay attention for that as well. Right, now you can see this lichen secondary metabolites. Actually, if you observe them under the electron microscope, you can see different crystal form you can see in the lichen thallus, right? So those are actually the secondary metabolites produced by the fungal partner. And they show some internal adaptations also, right? So I'm not going to explain it. And their chemistry is very, very important, right? Now, there are different techniques we can use for the identification of lichens. So one very easy techniques we call spot test, and the other one is the TLC. You know, it is most common for other plants as well, and the HPLC. And also we can use crystal arrangement and crystal shapes also to identify some lichens, because some lichens are there, they produce some crystals, those their shapes are very unique, right? So that character also we can use for the identification purpose. Now if I take this pathway, you know how they produce secondary metabolites. This algae mainly produce their glucose and everything, and it absorbed by the fungus, and after that those chemicals undergo different chemical pathways. There are mainly three pathways there: shikimic acid pathway, mevalonic acid pathway, and acetyl polymaltonyl pathway. So once they undergo any of these pathways, finally they produce their own secondary metabolite, right? So remember that always these secondary metabolites finally produced by the help of fungus, right? These are some of the group that they produce lichens, chemicals. So if I go to these different techniques immediately, now using we can use normal classic spot test, then microcrystal test, and TLC and the HPLC. Now in the spot test, actually basically we use three chemicals. One is 10% potassium hydroxide, that one we call K test, and normal sodium hypochlorite, or you can get some Chlorex available in the supermarket, that also used for the C test, we call C test. And there's another test we call P test. Paraphenylene, we use another chemical, paraphenylene diamine. So it's very carcinogenic. Whenever you want to use it, always you have to wear gloves and everything. Now you know that paraphenylene diamine is the main chemical that used to produce hair dye, right? So that chemical we can use for this spot test. Now what we can do is, if you found a lichen, even this can do even in the field as well, right? What you can do is, first you can add a small amount of 10% potassium hydroxide into one place, and sodium hypochlorite into another place, and paraphenylene diamine or P solution into another place, right? Likewise, now as the second step, what you had to do is, now we directly add that chemical onto the thallus, and the next step what we can do is we can remove the cortex, right? Then you can see the medulla. So you can see the medulla once you remove the cortex. Then you can do the same addition of the chemical into the medulla. Now after that, what you have to observe that whether there is any color change or not, right? Because those color changes also very unique for the identification purposes. There are some lichens, now as an example, when you adding potassium hydroxide 10% or the K solution, it turns into the yellow color, right? Or sometimes it turns into the red color. Likewise, due to some chemical reactions, you can easily identify whether that particular lichen is positive for the K test, C test, or P test. Based on that also, you can identify some characteristics of the lichen. Now you see, this is the C test. Now this is one good example. You see Now here this medulla is already open, and when you add this C solution or the Chlorex solution onto the medulla, now you see there is a proper color change. It turns into the red, right? That red coloration mean it has some specific chemical, right? So that that character we can use for the identification purpose. Now, when you go through the dichotomous key, in some cases it mention either C plus red or C plus minus. So that without delaying, I mean, when it's turned into the red, then what you can do is you can go through that pathway for the identification. If it is C plus red, if it is C minus, then we can go for the other pathway. Likewise, and this is the, and there's another one, K. First you have to add, just after adding the K you can add C, right? We call KC test. Again you can mention whether KC test plus color change or KC minus, and right, likewise you see this is the summary of that one. First you have to start K, add K, then K plus or minus. Likewise you can go through each of these spot tests, and finally you have to observe whether that lichen show any color change or not. Accordingly you can go through the dichotomous key and you can go for the identification purpose, right? And the microcrystal test also very important. Now here what you can do is you can take a glass slide, right? And after that you have to take a small piece of lichen thallus and you can keep that lichen thallus on the glass slide, and after that you have to add a small amount of acetone, right, onto the that small piece of lichen thallus. So then what would happen? All the secondary metabolites available in that small piece of lichen dissolve in the acetone and they come out from the lichen thallus, right, or the small part of the thallus. Then what you have to do is you have to you can remove the that small piece that you have already put on the or keep on the glass slide, and you have to keep it for a few minutes to evaporate the acetone, right? Then what you have to do, you can sometimes you better to cover it with the cover slip, and if you observe under the microscope you can see this very beautiful microcrystal arrangement, right? So that is another very famous microcrystal test. So because there are some literature, they have mentioned if this is the shape, then it has this chemical. If this is the shape, then it has this chemical. Likewise, by referring that book or the literature, you can get some idea which type of chemical available in the lichen, right? And the other one is the TLC test. I mean, many of you know about that. What you can do is you can usually use that small FN tube, and you can put small amount of lichen thallus, and after that you can add acetone, and you have to keep it for few minutes. Or if you can heat the FN tube, then you can speed up the extraction process in the acetone. Then after that, what you can do, either acetone or you can use any solution. Basically we use acetone, and after that you have to remove the you can take out the extracted solution into the capillary tube, right? And after that you can spot the TLC plate, right? And after you spotting that, you have to keep it dry. And there are different solvent systems available for the production of the lichen TLC, or to run the lichen TLC. So using that specific chemical, you can run this spotted TLC in the lichen tank, and after that what you can do is after run it to a top level, what you can do is you have to take it out. You can keep it for dry, and after that you can observe your produced TLC or the developed TLC under the UV light. You know, some many laboratories available UV cabinet, right? If you keep it inside the cabinet and if you observe at the top of the cabinet, you can see very UV active spot in the developed TLC, right? Then what you can do is you can mark them properly using a pencil where you spotted that UV active chemical, and after that you take it out, and after that you have to spray 10% sulfuric acid onto the lichen developed TLC, and after that you have to heat the TLC plate. Either you can use glass TLC plate or the aluminium coated one. In either case, after you spray in 10% sulfuric acid, what would happen, that sulfuric react with each and every chemical, and when you heat it, after evaporating that applied sulfuric, you can see different color patterns like this, right? So then what you have to do is you have to make the RF values, so you have to count the RF values and different colors and everything. There are some books already published with the different solvent system and different RF values, so that by referring that book, you can get a rough idea about what chemical present on the that extracted lichen, right? Likewise, now you can see in this image, there are more than 17 or 18 lichens spotted already, and only thing is that we have to spot another reference sample as well, because by applying reference sample, you can get some idea about the RF values, right? So likewise, you can use that TLC data for the identification purpose, right? So this is another one, and you can go for the HPLC techniques as well, because it is sophisticated and advanced techniques. I mean, in the TLC plate, you know, you saw different spot, no? But here, rather than spot, you can see different peaks like this, right? So, and another advantage is there, by calculating the area of this peak, you can get idea about the concentration of the chemicals as well. That means area of the peak proportional to the concentration of your secondary metabolites, right? So likewise, you can run the HPLC, then you can get idea. I mean, once you run it with the reference sample, what are the chemicals available, what are the quantities present in the lichen thallus, right? So those are the chemical tests you can use. So with that, I would like to conclude my presentation. Now there are so many resources available, especially if you go to the Google and search for any books, lichen keys, monograph, checklist, and also research article. So by referring those things, you can get more idea. Now I think now you know at least those haven't had a basic idea about the lichen. We discuss morphological characters, then anatomical characters, and after that chemical characters. So that just read a bit using different resources, then you can get more idea. And so with that, I will conclude my presentation. Thank you very much.
Thank you, Professor Jayalal. Thank you very much. Ladies and gentlemen, any questions to Professor Jayalal? You can raise your hand. I will allow you to speak. Okay, we have some questions in chat box. Okay, Pushpavati, please go ahead.
Sorry, I forgot to turn on my video.
No problem. Pushpavati, you can ask your question.
Yeah. Yes, sir. Nice presentation, sir. Thank you, sir.
Okay.
Sir, my question was about microbiome, isolation of microbiome. Can we isolate microbiome using the vegetative structure like the
Yes.
thalassem?
Yes. Actually, there are specific techniques available for the identification of microbiome. Actually, I am not thorough about that area, but there are different methods available. You have to use different filtration and filtering method. Using such a way, actually, you can separate them and even you can identify them, right? So if you can contact me, I will share my email, or I can send you more information related to that.
Okay, sir. Thank you so much.
Okay.
Professor Jayalal, Mr. Nikhil Verma had a question in the chat box. His
Okay.
question was, if a lichen is uprooted from its anchor, can it anchor again, especially for fructicose lichen?
Yeah, there are some research going on actually. They are trying to replant the lichens, right? In some cases, actually, they got successful results, but in some cases, they fail. Actually, still we don't know what are the other actual requirement for the regrowth of the lichen. We call transplant of the lichens. In many areas, scientists are doing such activities. In some cases, actually, they got positive results, lichen transplanting.
Okay, thank you. Lakshmi had a question. What is responsible for the flavor when lichens used as spices? Do you understand the question?
Yeah, many lichens, sometimes even in India, North India, and some China, they use to produce some flavor. So that is the character of a fungus, right? Now, thing is that because to produce flavor, they have to harvest in large quantities. So in that sense, it is in some way they harm the lichen community in that area, but they have some flavor. That the flavors always coming from the some reactions always incorporate with the fungus partner.
Okay, thank you. Professor Jayalal, Mr. Suraj Maurya had a question. His question was, many lichens are edible. How to identify edible lichens and what is their chemical compound?
So difficult question. Not all lichens are edible. I think I can't remember. There are some article published in India actually. They found, scientists not found whether they are edible or not, but the general people, because of their indigenous knowledge and everything, they were using such edible lichens for a long period of time. I think there is no proper mechanism to identify whether it is audible or not, but we can check for their edible characteristics, right? What is the component of protein, what is the quantity of carbohydrate and so on, but I have no thorough knowledge on that one actually. Good questions.
Professor Jayalal, Miss Sarah has a question. Is it possible to do cell culture for secondary metabolites through tissue culture? Do you understand the question?
Yeah, it is possible. Now scientists are doing such activities, but I have no idea whether they success or not, but they are trying to do such activities.
Thank you. Miss Shweta had one question. Can we use the crustose lichen for phytochemical extraction? Are there any methods to crustose lichens from its surface? Do you understand the questions?
Yeah. Thing is that actually when compared with the other types like folios or fruticose, the amount available in the natural environment is very limited, and also it's sometimes really difficult to take them out. But anyhow, what you can do is if you have any small quantities also, you can go for the chemical identification purpose. So once you identify the chemical, you can resynthesize in the laboratory. So that is the only possible way to get the secondary metabolites from the crustose lichens.
Okay, Mr. Tejas Kumar, you can ask your 3 questions now. Tejas Kumar, sorry. Yeah.
Sir, I would like to know if lichens have any specific lifespan period, and are they seasonal in growing or what?
Yeah, very good. Good question. Actually, there are crustose lichens. They have very long lifespan, so that in some time scientists use or they develop a specific area we call lichenometry. It's like carbon dating mechanism that because of their lifespan, scientists they were able to identify the age of the rock surfaces, right? But when compared with the folioses and fruticoses, they have high growth rate, and sometimes depending on the environmental condition, actually because of some other reasons, their lifespan may change, right? But anyhow, they can survive under harsh condition, as I mentioned earlier, because of the help of fungal partner. When they receive the favorable condition, so they can accelerate their growth.
Sheetal, you may ask your question.
Okay.
Sheetal, you may ask your question.
Yes,
Sir, one more small thing.
Tejas, can you please wait?
So my question is linked with the previous session,
Yeah.
that how we can differentiate between the prothallus, which is growing below the thallus,
Yes.
and the thallus, which is also growing below the new thallus because of trans-lichenization.
Yes. Now, thing is that now if they have prothallus, the one character is that color differentiation. Now, prothallus always black in color, and also it always grow much faster than the normal thallus, right?
Okay.
And other thing is that if possible, if you can take a cross section of that lichen, so then you can get a clear idea about the difference between prothallus and the normal thallus of the lichen. Now, thing is that sometimes you will mislead with the prothallus or some other substrate. So that only the way is that you have to take it and you have to take the cross section. Then you can see the link between the prothallus and the normal thallus, right? So that is a way to identify the prothallus. Most of the time, prothallus you can find with the lichens, those having cyanobacteria as the photobiont. Many such of cyanobacterial lichen have prothallus other than the other normal lichens.
Okay, so it will come with the experience that we can
Yeah,
identify.
sometimes, yeah. But at the beginning, it's really difficult. But I mean, in case if you found something, please just let me know. So I will help you.
Yeah, thank you. Thank you so much, sir.
Okay, welcome.
Mr. Tejas Kumar.
Sir, sometimes we might not be able to find out what are the species by using the chemical or anatomical characters. We cannot find out. At
Yeah.
such, what molecular level techniques can be used, and for a researcher, what will be the financial cost for using such techniques, sir?
Which technique?
For molecular techniques,
Okay.
they might.
Yeah. Yeah, the thing is that actually, I mean, just imagine if you completely identified your lichen using your morphological characteristics, but we always prefer to go for the molecular characteristics as well. Nowadays, without having having only the morphological characters, most of the journals not accept it as the new species, right? Therefore, we have to always go for the molecular level. The only thing is that we have to extract the total genome. Usually, we use this ITSR1, ITS region, and there are some other regions also you can multiply. And considering cost, actually, I mean, you can do your PCR or you can make your PCR product at the laboratory level, but only thing is that for the sequence process, you have to, I mean, if it is available in your university or your country, you can send them to that institute, or otherwise we have to send them to another country. So it takes, I mean, you have to pay some money for the sequencing process. So that is, I mean, and also sometimes, I mean, rather than different chemicals, we can use some kit, right? That mean all the chemicals are in a single bottle or something like that. So that, however, if you go to the molecular level, it's somewhat costly. I have no idea in the Indian situation, but in Sri Lanka, we have only few places available for the sequencing. Anyhow, we have to pay for sequencing process. And also, sometimes it is, I mean, more worth to send abroad to sequence the sample.
Thank you, Professor Jayalal. Tejas, Mr. Tejas, in India, we could do the just sequencing of the ITS for 1,000 to 2,000 rupees by some using the services of some private companies.
Sir, are there any such contact for such companies?
Sure, we'll see. I will leave my mobile. I will leave my mobile number in the chat box. Please contact me. Okay. Are there any more questions to Professor Jayalal? Mega, please ask. Mega, would you like to ask the question? Okay, Mega has this question, Professor Jayalal. Have lichen developed any coping mechanism to survive environmental stress? If not, do lichens get extinct without any survival mechanism adapted?
Yeah, I mean, like all the other organisms, they also try to make copies to survive in the environment. But apart from the other organism, actually, lichens are very unique. Why is that? I mean, not all the lichen, but some lichens, they can tolerate very, very harsh conditions in the environment. So that is the specificity of the lichens.
Professor Sarma, would you like to ask the question?
Yeah, it's a wonderful lecture, Professor Jayalal.
Thank you.
I have a question. How easy it is to differentiate between lichenized and lichenicolous fungi?
Yeah, good question. Now, thing is that now what you can do is now, as I mentioned earlier, if you pay your attention for the lichenized fungi, they always present inside the thallus, and first you have to remove the lichenicolous fungi. And or other way around, what you can do is now when you closely look at of the surface of the lichen thallus under the stereo microscope, right? You can see that they produce especially that pycnidium or the pycnidial structures. So that is the easiest way, I mean, to identify whether any lichen consists such type of any lichenicolous fungus or not. I mean, using morphological characters, easily identify. I mean, you remember I mentioned that sometimes some tiny dots are present, or sometimes like that, or sometimes they deform the lichen thallus, like galls. I mean, because of there are some activities, they form sometimes gall-like structures, right? so that when you observe them in the under the stereo microscope, that is one possible way to identify Lichenicolous fungi.
Professor
Thank you very
Jayalal.
much. Thank you very much.
Professor Jayalal, I have a question. Do you have a national facility in Sri Lanka where the lichen identification can be done on payment basis?
At the moment, we don't have, Dr. Sinol. But what we do is we just contact the experienced person, and usually they do that process.
Okay, thank you. And Nikhil Verma has the question. Has a question. This will be the last question. Yeah.
Thank you. I hope I'm audible this time.
Yes, you are. Yeah.
Thank you very much. Sir, I would like to know how lichens grow in nature, both on rocks as well as in trees, because on trees, I see large clumps sometimes falling and you can pick them up when you go and trek in southern India. On rocks, also you see big circular formation. They would be as big as 10 centimeters and more. Also in Himalayas, you see them, but the presentation, like you said, and I also realized that they grow probably only 1 or 2 millimeter per year.
Yeah.
So is that in lab, or they all grow only with that small? Because that will be too limiting for a species to survive, I feel.
Actually, it depends on the species. Actually, there are some species, especially the crustose species, they show very slow growth rate. Because of that slow growth rate, whenever you observe that thallus, always you can see the small thallus. But however, there are some foliose lichens, like you may have seen that very large patches available, right? If they are present on most of the time fully exposed rock, you can see very luxurious growth on the rock surface. So that growth rate actually mainly depend on their genetic structure. However, due to some environmental factors also, you can get either luxuriously grown lichen or not. Now, sometimes in the same lichen, if it is in the shady environment, sometimes you can't see that much of growth rate. But if it is exposed to enough sunlight, then you can see the luxurious growth. So it may vary actually depending on the environmental condition and also the genetic factor.
So the large ones could be few 100 years old?
Sorry?
Would the large shapes mean that they are few 100 years old?
No, you can't say like that.
Okay.
Yeah, you can't say like that.
Professor Jayalal, one more person has raised her hand. Can we take the question, please? She's from Sri Lanka.
Madhara.
Yeah, Madhara. Madhara, please ask your question.
Thank you so much, sir. This is a very informative presentation. So I have one question regarding to what are the industrial application and bioremediation applications of the lignocollus fungi?
Yeah, good question, Madhara. Actually, they have so many industrial applications. I have not mentioned them in this presentation. The one thing is that the secondary metabolites show so many antimicrobial activity, so that even in India, they have made so many pharmaceuticals from the lichens, and also they show they have some specific dyes. Now, as an example, litmus we use for the production of the litmus, that dyes coming from the lichen. And also in large garments, right? They use to make some unique colors from the lichens. Likewise, there are so many, industrial applications are there. And considering the bioremediation, actually, nowadays some are doing some researches because, even if the lichen present on the natural environment, due to now it is found by some scientists. Now with the rain, what would happen? That secondary metabolites, not all metabolites, some metabolites get dissolved in the rainwater and they come down to the bottom of the tree. Then what would happen? Because of their some biotic activity or remediation activity, they vanish some weeds just around the trees, right? So that is a good sign that that lichen some lichen species can use as a good bioremediation, but many of the studies are still going on in the laboratory scale.
Ladies and gentlemen, I need to stop this session now. Sorry for that, but Professor Jayalal is available in our WhatsApp group. You can always pose a question to him in the group. I think he'll be very happy to answer such questions on lichens. Yeah. Over to Romana for the vote of thanks.
Okay. Good afternoon, everyone. Thank you, Jayalal sir, for sharing vast knowledge on identification of lichens based on the morphological and anatomical features. I thank Shinoy sir for organizing such an interactive session. And lastly, I thank MycoAsia team for the kind cooperation. Thank you, one and all. Thank you, sir.
Thank you, Ms. Romana and Dr. Shenoy and all the participants. And thank you very much for giving this opportunity to share my knowledge related to the lichen. I hope at least few of you continue with the lichen research work. So that would be, more grateful for me. Thank you very much.
Thank you, Professor Jayalal. Hope to see you soon somewhere in India or Sri Lanka or
Sure,
somewhere
sure,
else.
sure.
Yeah.
Thank
So
you.
much. Yeah.
Okay,
Bye. Thank you, ladies and gentlemen. Yeah. Bye-bye.
Bye.